Everybody. Welcome to our Earth Journalism Network webinar on zoonotic diseases. Thank you very much for joining us today. My name is James Fahn. I'm the Executive Director of the Earth Journalism Network. And first and foremost, we'd like to wish all of you a very happy Earth Day. We do realize it's not the happiest of times for our planet and its people, but let's all give thanks for the life we're able to celebrate, and especially give thanks to the folks in the medical community and all the people working on the front lines in essential jobs to keep us healthy and fed and informed, because of course, many of you journalists out there, you also are working in essential jobs, and we very much appreciate your work keeping us all well informed about the pandemic and everything related to it. We are very happy today to be joined by a team of researchers from the USAID PREDICT and PREEMPT projects. They're working to detect and contain viruses out in the field, and I'm sure they have a lot of interesting experiences to share with us. I'm gonna turn it over to them in just a moment, but first, just a few words about the Earth Journalism Network and who we are and what we do. In case you're not familiar with us, we are a project of Internews, a global media development organization, and we're also a community of over 12,000 journalists from more than 180 countries who are all dedicated to improving the quantity and quality of environmental coverage around the world. You can find out a lot more about us on our website at www.earthjournalism.net. And certainly encourage you to check that out. And if you're a journalist, feel free to join. You can register on the website. Registration is free. We have a lot of activities and opportunities that can help you do your job. This webinar is one of a series of webinars that we are holding about zoonotic diseases and about their environmental origins, particularly in the wildlife trade and other forms of ecosystem disruption and that are helping or causing a lot of viruses and other pathogens to spill over into human populations. The plan for this webinar is it will last about an hour, 60 minutes or so. Half of the time will be spent with a presentation by our colleagues here, our panelists, and then we're going to try and give you a good amount of time for questions and answers. We definitely encourage you to please submit your questions. You can do so using the Q&A feature. You'll see that at the bottom of your screen on the platform. You'll see the Q&A. Please do enter your questions there. And if you don't mind, please just let us know who you are, your name, and where you're from. That's always nice to see. I realize there's also a chat feature using platforms, but we'll try and use that for other forms of other communication. So please do look in the Q&A section for questions that are being asked, and feel free to ask your own too. We're going to post a recording of this webinar on our website at earthjournalism.net, so you can certainly check it out afterwards, and feel free to share it. We certainly encourage that. And finally, just to mention that we have a bunch of upcoming webinars on related topics over the coming days and weeks. Tomorrow, there's going to be a webinar sponsored by our colleagues at the East Africa Wildlife and Conservation Project. So that will be a webinar on the wildlife trade in East Africa. You're welcome to join that. That will take place at 3 p.m. Nairobi time. Next week, and in the following weeks, we'll have further webinars looking at how to report on the wildlife trade, how to do investigative reporting, how to do data journalism regarding the wildlife trade. And a lot of those will be focused in Asia and the Pacific region. We'll have journalists from China, Vietnam, and the Philippines as speaking about how they go about their work. And again, you can find all this information on our website at earthjournalism.net. But without further ado, I'm going to turn it over to our panelists. I'm not going to introduce all of them. I'll just turn it over to our moderator, David Wolking. He's with the One Health Institute at the University of California, Davis. And he's going to introduce the rest of his panelists. And thank you again, ladies and gentlemen, for joining us. I'll turn it over to you, David.
Yeah, thank you, James. Thanks everyone for joining. We're really excited to be with you all and to hopefully give you a nice tour of some of the work we've been doing with the USAID PREDICT project. We're not going to get too much into the PREEMPT project today, but it's largely a lot of us working on similar efforts. So James Mangura and Sierra Leone and I work on PREEMPT together, but we wanted to focus on PREDICT, I think today predominantly because it has a larger geographic footprint. So PREDICT is a pretty big program, working in over 35 countries over the past decade, and is winding down. So we're in our last 6 months of the project right now. So I'm gonna share some slides. And as I hand it over to each speaker, I'll do a brief introduction of the rest of the panel. But just to get us started today, let me get these up. Okay, so we're going to get into how the PREDICT project in particular works around the world to build capacity and really strengthen capabilities to detect and prevent viruses from emerging. And how do we do that? Where do we work? So we tend to work in hotspots for virus emergence, and we sort of start with the math. And Dr. Pranav Pandit is going to speak a little bit more about using math and the data and modeling tools and techniques to really inform decision making and help us guide surveillance and disease detection efforts. But we work with PREDICT. We started with some models that showed us where some of the hotspots for emerging viruses really were around the world. And a lot of these were in low and middle income countries. And as a project funded by the U.S. Agency for International Development, we worked with a lot of these country partners to identify areas in countries, in Latin America, Africa, South and Southeast Asia, that were, that had a lot of what we consider drivers of disease emergence. So areas where there's high levels of biodiversity, population density, and a lot of landscape change. So things that come together almost in a perfect storm to, allow or enable a virus or a disease to spill over from wildlife into human or domestic animal populations and then eventually spread. So in how we work, we work in partnerships with local governments and scientists and universities around the world. And Dr. Grace Mwangoka will tell you a little bit about how this works in Tanzania in a little bit. But our approach was to use One Health. And that's sort of an idea that the well-being of humans is inextricably linked to the health of the planet. And recognizing that, the health of humans and wildlife and animals and ecosystems are linked. And to really understand sort of these challenges and address these big challenges like, virus spillover or pandemic threats, you really need to build big teams, multidisciplinary teams of experts. And I think the panel today is a pretty good, mix of individuals with diverse expertise. Grace is a veterinarian, James is a public health expert, Pranav is a bioinformatician and a mathematician. And so bringing us all together on teams to work on these problems is really how the PREDICT team kind of built a really interesting platform and network that allowed us to do some great work. So this is just a really quick concept slide to show you how we put our work in action in some of these areas. And using this One Health concept, we used One Health to create sort of a surveillance design that would take our multidisciplinary teams and go out into these hotspots for virus emergence. And really target some key interfaces. So interfaces are basically areas where, we've identified, high risk for contact with, wild animal populations or, basically the ingredients to create a viral spillover, recipe. And we would go out into these communities and work with community members to really understand what were the human behaviors and what were the factors that could be, linked to a virus spillover event. And then with their permission, we would go out and collect samples humanely and safely from wildlife populations and partner with health centers and clinics and hospitals, and even, in the community themselves to collect samples from people. We would take, our team. So this is a sort of a photo of this One Health work in action. This is a team in Nepal working at one of these, high risk interfaces that we identified in an urban area of Kathmandu where there's a lot of informal settlements. So just settlements of people sort of growing up in a rapidly expanding city with a lot of human population movement. And, the dominant wildlife species in these areas was often, rodents and bats living in and around people's homes, and, also non-human primates like rhesus macaques that are pretty prevalent in that area. And so our teams would, get into their protective gear, to prevent themselves getting infected in the field, and safely capture wildlife and collect samples and then release the animals back into the wild. So a lot of our PREDICT work is very conservation focused. So we believe very strongly in protecting and working safely with wild animal populations. And that same team in Nepal and everywhere else we've worked in PREDICT would partner with the local health centers and community members to understand their risks and their health concerns and collect samples from the human community as well. And what do we do with all of those samples? Well, we worked also with laboratory networks in each country, and we created a disease detection platform that had the capability to screen samples at the viral family level, which lets us cast a wider net. So instead of testing for a specific virus, like an Ebola virus or, SARS coronavirus 2, for example, we would test for filoviruses, the family that Ebola belongs to, or coronaviruses, the broader family that, SARS-2 is a part of. And that lets us, gives us the capability to detect, not just these known pathogens and known zoonotic diseases, but also the potential new emerging threats that might be circulating in these wildlife populations or even in the human communities themselves. So we worked with laboratories around the world in over 35 countries, more than 60 labs so far, to build that molecular virology capacity for virus detection, and then also, help sort of train and equip the laboratory workforce to do this work, independently in their labs, and also work to really create a sort of a One Health laboratory network where we'd try to twin the veterinary and public health laboratory systems together for information sharing and connect them to a big global network of disease detection experts. So there was always, the capability for mentorship and information sharing, and really sort of strengthen the entire system that way. We also launched some behavioral risk investigations. So partnering with social scientists and behavioral scientists, we put in place some epidemiological and behavioral scientific research methodologies to really understand, in these human communities, what are the factors that could be linked to a virus spillover event or causing viral transmission between animals and people. And this work really allowed us to do some deep dives into particularly at-risk interfaces. Interfaces like the bushmeat value chain in Central and East Africa, wildlife markets in South and Southeast Asia, the informal settlements I was talking about in Nepal, and ecotourism, and other at-risk interfaces like back guano farms and things like that. And I think Grace will probably get into some of those, and Pranav as well, in their presentations. And what do we do with all this data? So once we, have some virus findings, for example, in Sierra Leone, we detected a new Ebola virus called the Bomboli virus. And our teams detected that in bats, and James Bangura was, leading that team. And they detected this virus in bats that lived in people's homes. And we needed to really rapidly put together a plan to share what that meant for these communities. These bats were, in the communities, living in their homes. They, harbored a new Ebola virus that had never yet caused disease in the human population that we were aware of. And so we worked with the government of Sierra Leone to design this behavior change and risk communication strategy that we entitled Living Safely with Bats, which was a pictorial book. This is just one example of these communication tools, but a pictorial book that the team in Sierra Leone could take to the communities and talk about the risks of bats, but also the benefit of bats to ecosystems and ecosystem health. And so we've got, resources like that now translated into lots of different languages and freely available online. And this slide showcases a few of the other tools that we put together, some in Swahili for the East Africa region and in French for West Africa. So I'm going to just give a quick snapshot of PREDICT's impact before I introduce the rest of the panel. But over the last 10 years, PREDICT has trained more than 6,800 people, collected samples from hundreds of thousands of animals and people, strengthened the capacity for disease detection in more than 60 labs, and we've detected and discovered 1,000 plus viruses, as you can sort of see there. And what does this mean? I mean, this is really what PREDICT was working to do was strengthen the capability of, some of the areas, some of the countries and the systems and the most vulnerable areas for disease emergence to be ready to, respond to a disease X, which this time around was COVID-19, and sort of a proof of concept. So it was a feasibility assessment to find viruses ahead of an outbreak. We determined that emerging threats can be identified before spillover, for example, with the Bomboli virus in Sierra Leone. We also developed some financial projections to forecast. So how much would all of this cost? And, worked with some teams of economists to really understand the cost benefit to, preventing a pandemic, to preventing outbreaks, to doing sort of this work up front. We've identified and trained, qualified professionals and really built this One Health workforce, which is now standing ready to help out in any emergency situation. And even with COVID-19 is called into action and, working quite a bit. And we can talk about more of that in the Q&A later. We've, worked in over 60 laboratories, and we've cataloged these high-risk viruses and the metadata associated with them. So really trying to understand the risk for mitigation and what we can do about them. And I think Pranav's going to give a really nice overview of that in his presentation. But perhaps most importantly, I think we demonstrated the importance to really work together cooperatively, globally, and in a multi-sectoral One Health way to, on emerging infectious disease control and prevention. So with that, I'm going to turn it over to Dr. Grace Mongoka, who I've been working with for the last 6 years, and I've had the pleasure to partner with her in Tanzania. And she's going to show you a little bit about how PREDICT Tanzania was put into action, how we put this One Health approach in action. So Grace, you're welcome.
Thank you, David, for a very nice summary. In Tanzania, my name is Grace. Like David said, that during PREDICT implementation in Tanzania, I was coding the human surveillance and part of the behavioral risk assessment. So in Tanzania, we had to implement PREDICT in One Health, putting One Health in action. That our institution, which is primarily dealing with the human health, we had to coordinate with the Sokoine University. Really dealing with it. The next slide, David. No. Next slide. So in Tanzania, we had a community surveillance, and this was primarily being done in the northern west part of Tanzania. And this site was selected based on they are located at a border bordering Uganda, Congo, and Burundi and Rwanda. And most of the time there's uncontrolled movement of people, and some of the areas they have in one of the sites, we have the larger refugees camp. So there is quite a big interaction of the community themselves and the people around that area. So we had concurrently sampled the human and the wildlife in close proximity, just making sure that we capture any viral transmission, what can happen within a species, but if they can happen, they can spill over to human. So we had one component on the clinics. So we collected samples from the human at the health facilities, but also we collected samples from the community. We are assuming that most of the time when people are sick, they don't go to the hospital or they self-medicate. So to get those that they don't go to the hospital, we had to collect the samples from the community. And these communities we selected based on their livelihood. Most of them, we feel that they are more at risk of getting the infection. But in parallel to that, we had the questionnaire, which we have the survey, but also focus group discussion, where we have differently asking people about their awareness. But again, if there's any biosecurity measures that have been implemented to prevent getting them infection from their animals, being livestock or wildlife. And some of them also the disease transmission and zoonotic diseases in their community. And this surveillance took place during the wet season and dry season. It was important to make sure that between the two seasons. So from the community and even the health clinic, the biggest challenge was to make sure we get a viable sample. So we had to, most of the time, travel like more than 1,000 kilometers to refuel, especially for health clinic with the liquid nitrogen and supplies for all because we are working with the healthcare workers and get a lot of support from the government too. Next slide. Next slide. So from the community, we had to transfer all the samples to the lab. Like David mentioned, that building capacity to our laboratory was very, very critical. And like now we see the potential of what PREDICT did, making sure that the local laboratory have the capacity to detect viruses. So for PREDICT, we had the opportunity to get a mentorship of a senior laboratory scientist from UC Davis. Brett, who came to mentor the two laboratory, and they had an opportunity to test 5 viral families. This we call the big 5, which have the potential of causing pandemic. And now we have the COVID-19 is within the coronaviruses, but the capacity has to expand. So the two team from the SUA and the IHI has to transfer the skills and knowledge that they have to more local people, meaning the intern, the student, even the government laboratory staff. The picture on the right, because we had the workshop where the government staff, they had to come to attach to our lab, at least for 2 weeks, to get a hand-on experience on testing these viruses. So we had to make sure that the staff from the human lab, the wildlife, and livestock come together, and also to discuss some of the challenges that they found out in their working places, how they can share the information, the protocols, and improve their knowledge too. Next. Next slide. So from the community, from the laboratory, we had to go to the community where we had to really inform them about what we found out. We had the community engagement from the beginning. We had through the community meetings, but during the sampling, we continue with the discussion during the survey. But from this, the result from the PREDICT was very eye-opening, where we confirmed that bat really the host of many pandemic viruses, including Ebola, SARS, and MERS. So using the same bat book, which is developed by our colleagues at the PREDICT team, we had to translate it in Swahili, because during the surveillance, we had experienced more of people had in contact with these bats, especially in their households, but some of them, they eating the bats, some of them even fresh. They are harvesting bat guano for fertilizers. So we had to, when we got to these findings, we had to go back and try to educate them. Of course, the community was very engaging, and some of them, they are offering some local method that they've been using to get rid of the bats from their households. But some of them, they've changed the behavior, meaning they have stopped eating the bats. So, but the challenge is changing human behavior is a process. So we had some of the material that we have to leave behind to stick them in maybe in the market, in different hospital or health facilities, for them to continue looking at them and observe. And I'm sure by now they remember what we told them about the risk of this getting infection from these animals. So we had a chance of getting not a very large number of people, the community leaders, and there are representatives, the key stakeholders from the local governments, but even individual communities also attended in this meeting where we involved them. Thank you. Maybe I'll talk more during the question and answer.
Yeah, yeah, thanks, Grace. I think it'll be great to hear some questions from the audience later on about the work in Tanzania. But now let's turn it over to Dr. Pranav Pandit, who's a really great mathematician who's been working a lot on integrating data from both PREDICT, but also open data sets around the world, and trying to really use some of, the modern data science tools to help inform how we do surveillance, how we identify these hotspots, and also, how we use that information strategically to move towards prevention and towards, identification of intervention strategies and even countermeasures. So Pranav, welcome, and thanks for joining today.
Yeah, thank you for having me, and thanks, David, for the introduction. You just heard from Dr. Grace about how PREDICT is working in Tanzania. So I think now it's a great time to move and look the project from slightly a global perspective. And within this presentation, I'll try to explain how the vast data the PREDICT project has been collecting for last almost 10 years. We are using that to identify and develop models that can help us determine policies so that we can mitigate risk related to emerging pandemic threats, especially from viruses. So if you go to our next slide. Before we go into any specific models, I just want to reiterate the PREDICT surveillance strategy. As Dr. Grace and David has already mentioned, it's a risk-based surveillance strategy. What it means is that we have been targeting animal taxa that are known to live in a very close association with humans, which include bats, non-human primates, rodents, different birds, of course, domestic animals, and some other wildlife taxa that are known to stay along with people. And we are also trying to identify viruses in key viral families, which include coronaviruses, paramyxoviruses, filoviruses, and flaviviruses. All of these are known to cause diseases of public health importance, and of which, as you know, coronaviruses is right now one of the viral families of great concern. But other viral families, including flaviviruses, caused significant public health outbreaks about 4 years ago. Filoviruses, which includes Ebola viruses, are also known to cause outbreaks in humans. And here you can see just the timeline of our surveillance efforts. And over the last 10 years, we have sampled more than 100,000 individual animals from wildlife, as well as about 30,000 people. And we tried to detect these viruses from these 5 viral families. And we have, until now, we have detected almost more than 900 completely new viruses during this surveillance. And it's just not the data about new viruses, but all the metadata, that ecological data associated with those animals and those viruses that we have been collecting. And we have been using this data sets to develop our models and ascertain the risk related to these new viruses. So within this presentation, what I'll do is basically show you a few examples of the models that we have developed, which first try to identify the key human-animal interfaces that we think are important in disease transmission from wildlife to animals, and how we use those models to identify key species of wildlife so that we can develop studies related to those species and understand the ecology of those viruses in detail so that we can develop policies to mitigate threats from those specific species. So if we go to our next slide, we'll have our first example where we try to identify key animal-human interfaces. Here in the plot, you can see that on the vertical axis, there are species that have been classified according to IUCN threatened status. And the bubbles basically represent the number of viruses that those groups of species share with humans. And as you can see, the bigger bubbles represents higher number of viruses. And you can see that species that have been threatened due to human exploitation, especially due to exploitation of their habitats and just exploitation due to hunting, tend to have higher number of twice as many viruses than other species. So what it means is that it indicates us that these are the key animal-human interfaces, which includes live wildlife markets, bat caves, that needs to be protected. And that's where the transmission from wild animals to humans tend to occur. So this is a recent study that we published in Proceedings of Royal Society B that is talking about our new results here that identify key human-animal interfaces. So the next example I'll give is about flaviviruses. If we move to the next slide. So quick introduction about flaviviruses. If you remember, about 4 years ago during the last Olympic year, there was a big outbreak of Zika virus that happened in Brazil. And Zika virus is a part of a bigger viral family called as flaviviruses. And it also includes other flaviviruses such as yellow fever virus, dengue, West Nile virus. All of these are viruses of public health importance, and they are spread globally. They are everywhere on all the continents. And we wanted to see what are the key wildlife species that we should target so that we can develop the surveillance around these flaviviruses, and hence this model development happened. So all of these models, how they work is that we try to look into known hosts or known knowledge about these viruses and learn from that knowledge and infer upon the emerging viruses from those viral families. So what we basically did was we looked into the data and saw what are the species, what are the bird species, what are the primate species, what are the bat species that these viruses are known to infect. And here in the dendrogram, in the clustering figure, you can see that that some viruses tend to infect similar type of hosts. So like, for example, next to the primate icon, you can see that Zika and yellow fever tend to infect primates. There are some viral species that tend to just infect bats. But then there are some viral species that tend to infect wide variety of animals, including birds, horses, and humans, of course. I mean, all of these are zoonotic viruses that tend to infect humans. So what we do is basically we develop models that learn from this data and try to predict what could be the missing hosts of these flaviviruses. And here you can just see in the maps below is the distribution of predicted hosts for these flaviviruses. The first plot basically shows the distribution of yellow fever and Zika hosts. The red area, the higher diversity of predicted hosts. Similarly, the middle plot shows the distribution of dengue hosts, predicted dengue virus hosts. And then the last plot shows the distribution of Japanese encephalitis hosts. So this is how these models are helping us not just identify species, but identify regions where we should go out and do more sampling and do more studies. If we go to the next slide. So now we have seen how these models are inferring upon known viruses, known emerging viruses. But what about predict viruses? Can these models help us tell anything about newly identified predict viruses? And the answer is yes. So the way we are handling this issue is we're trying to understand the community of viruses and their wildlife hosts. So if we just look at the network on the left side, basically each dot in the network represents a virus. And when and those two viruses are connected to each other when they share a wildlife host. What it means is that both those viruses are found in the same host. So on the left side is a network that is generated based on the knowledge of all the viruses in wildlife. That's how the structure of their sharing a host looks like. And when we develop those models based on these networks and we add our novel viruses that we have discovered in PREDICT during the last 10 years, you can see that these PREDICT viruses represented by white dots in the right figure basically where do they lie in the network and what is their position in the host network of this virus host community. And this representation of the network helps us identify what are the new PREDICT viruses that can infect wide variety of species. what are the new predict viruses that can just infect some type of species. They are very well specialized to certain types of species, while some might be just found in only one species, and they are very super specialized to only one species. So these network models are helping us develop further about the ecology of these newly discovered viruses. If you go to the next slide. So why are we developing these models? Is first of all, this is to develop a targeted wildlife surveillance for emerging infectious diseases. As we have been listening about from David and Grace, that we go out and we try to identify hotspots where the transmission from wildlife to human can occur. But to identify those hotspots, these models are very essential. For example, just take an example of Zika virus. Before the modeling work, we just knew about few species that could infect Zika and yellow fever, let's say about 14. But now we have a list of about 112 species that potentially could harbor Zika virus. And probably we should go out there in wildlife and sample all these species. And not just the list of species, but what type of animal taxa we need to target is also informed from these models. And as we saw in the maps, we also know where these species tend to exist. So we can also identify geographical hotspots. If we go to the last slide. So I want to, in this last slide, I just want to mention here that the whole modeling is an iterative process. So we develop models based on the available data that we have and use the inferences from the model to do further fieldwork. The fieldwork basically then generates new data, which eventually again helps us improve our models. And within this circle, within this iterative process, every time there is a policy decision that needs to be made, these models are helping us develop those policy decisions so that we can mitigate the risk better. So eventually, since it's a very iterative process which requires multiple expertise, it's a big teamwork. And here is just a photo of the whole PREDICT team all together with all their different expertise working together to develop this project. So with this, I think I hand it over back to David. Thank you.
Thanks, Pranav. I think we're sort of at a point with the PREDICT project now where we're using a lot of the data we've collected over the past decade to work through it, analyze it, interpret it, and really come to some key insights on what it means for predicting viral spillover risk and what we can do in terms of strategies for interventions and prevention. But we wanted James to talk a little bit about today, James Bangura, what happens when you don't get there preemptively, when you after a virus spillover event happens. And James has had a lot of experience in Sierra Leone working in outbreak situations. He was part of the team working on response during the West African Ebola epidemic in Sierra Leone. And he's now working in the Situation Room and with the Outbreak Task Force in Sierra Leone on COVID-19 response support. And so we just wanted to give James the opportunity to talk about that side of the story as well. I mean, PREDICT is, our primary mandate has always been to, work upstream of any virus spillover and spread event. So before an outbreak and really understand the threats, pre-emergence, so to speak. But because of our capability and our One Health approach in our teams, we also have a range of experience working and supporting our host country governments with outbreak response and with technical assistance. And that's something that James does excellently. So James, over to you, please.
Thank you, David. Thank you very much. In Sierra Leone, what we are doing, we have moved from the routine activities that we've been implementing to reacting to the spillover now. We have worked with the government for the past 4 years on preventing or working hard to detect spillovers. Now we have spillover. When the COVID-19 pandemic started, we changed our services, our programs to supporting the government to prepare. and putting systems in place to detect any spillover or any importation of the virus into the country. We moved from preventing to detect now to responding now, building capacity to cope with what we are experiencing now, from working with the national government onto district, that is sub-national government, to prepare systems, building teams to respond adequately to the pandemic. Yes, next slide. Next slide, please. So at the time of the preparation of these, not the previous slide, please. Thank you, that's the one. At the time of the preparation of the slides, we, at this point, the country had reported, had cases, sporadic cases in 4 of the 16 districts that we have. Mostly the cases are coming from the city, the capital city, which is Freetown. And of those cases, we've seen over 39% of these cases are occurring among healthcare workers within Sierra Leone. And the system that we are working and supporting, we moved from the surveillance to look for the virus in animals to now building systems to implement a system to detect the virus into humans, like estimating who is exposed within the communities. So primarily we are using an event-based surveillance system, which was built, from the 2014 Ebola outbreak. There's a toll-free line, which is the 117 line, which is populated with well advertised across the country. The people who, whatever events of travelers into the country or unusual occurrences across the country, or individuals that are sick, could be your relative or could be at the health facilities, everyone is encouraged to call on 117. Is a toll-free one to notify the event. So the the system is built on when those alerts are sent in, the 117 has a 24/7 staff that logs in these alerts automatically, and then they push them into the surveillance system for case investigators to do the investigation. Next slide, David. Before we had the first case, we worked on with the national government to put system in place. How would we pick up the first importation? So the photo you see in the early days before we had any first case here in the country, working with the districts, the man with the black shirt there that sat down is the district medical officer for one of the border districts, having systems in place to having meetings with the border districts, putting systems to pick up any importation. Even before Guinea had, and then before Liberia, our neighbors had any cases, these structures have been in place to guide them what we need to do and what event that needs to be investigated, what constitutes an alert that needs investigation. And then all of those were built upon the 2 approach that helped us to pick up the cases that we're having now. The verification of all the alerts that I mentioned earlier that are coming through the 117 system. We work with the government to be sure that we have individuals at health facilities that will investigate alert individuals that either they walks in or individuals that were alerts are sent by the contact tracers in the various communities across the country. Wherever contact we have, those people who we have trained to make sure they conduct comprehensive investigation of those alerts. We also have a second approach that we're using is finding the cases. Apart from the events that are being reported onto us, we work on building teams with the government together with other technical partners, the WHO, the CDC, and MSF. We work together, training personnel and then healthcare workers, medical students, medical graduates, to go into these communities, like identified hotspots where cases are coming from now, to and investigate individuals that could be at risk. we establish sentinel surveillance at high risk, those communities. We have identified pharmacies, healthcare attendance has gone down, like clinic attendance has gone down. But we have built a system wherein those that walks into the clinics can be investigated, and as well as also going in retrospect, reviewing clinic registers for individuals that have come and then presenting flu-like illness that are mimicking to COVID-19, and then those are all being investigated through the Sentinel system, and as well as the pharmacies as well. Many people will just walk to a pharmacy and then get over-the-counter medication, which is not very well restricted here, and then they get medication. Those are individuals. We have a building system with the government to make sure with the pharmacies and then the structure that the government have to make sure that those individuals alerts are triggered so we can investigate those. And then in the high volume facilities, again, in the communities where we are having cases, we've also worked with the government to put system in place to be sure that when these individuals are being seen, we can also prospectively investigate them to see what they have over the period, again, up to this moment. Next slide. So the way the system works, the event-based system, it has a flow. So these alerts are triggered by either the community members or from the health facility, or it could be a friend, or it could be individuals who trigger this. They are generated at the 117 system. We have various teams that we have worked with the government together with other partners, the WHO, the CDC, to train individuals we call case investigators. These are people with health background, could be most are nurses, some are medical graduates, house officers, and then some are medical students in the final year of medical school, with whom we have trained as case investigators to investigate all of these alerts to be sure that these alerts meet the case definition. Once they are given this alert, they move to the various locations where the alert comes in from, and then they investigate. Once the individual meets the criteria that will that we look for to suspect a COVID case, and then the lab team is notified here. The lab team goes in, collects sample, and the individuals are isolated. We've also had situations wherein we train these individuals to be able to identify some alerts are primes, and then some are also related to non-COVID. So also looking through, we refer to them as false alerts not related to COVID, but if they have got their non-COVID illnesses, also building a system wherein they can be cared for outside the COVID response structure that we have. This is the main structure that fits in, and then that facilitates the surveillance for within the country right now. It's like a surveillance system. We move from surveillance system in animals and now a surveillance system in humans for specific events related to COVID. And that's the flow on how the system works here in country. And this has been very successful thus far in helping us pick up the cases that we have. Next slide, please. Next slide, David. At the moment, in the country, we are having clusters of cases. So within these clusters of cases involve engagement. We work with the military, you see the photos, and then also work with communities, engaging them to carry out comprehensive investigation of every case. We're at the point where we have the first few cases, and within us as a program, UC Davis and WHO, CDC, we sit together with the technical partners at the technical meetings every day. We look through case by case that we are having. We still have the opportunity in country, and then look through what needs to be done. And then after focus is mainly identifying, trying to connect the transmission, identifying how did this individual got exposed. And then a lot of intervention, a lot of steps are involved in this, engaging communities, talking to community members, talking to maybe pharmacies, whether they could recall on what has been happening over time before we had a system in. And then also, of course, be walking through these high volume facilities. And then at times again, you see on the second photo below, you'll see meeting with community leaders to talk through, this is what we look for. And then to give us the stories we're interested in, understanding what is happening in those communities. And then that has helped us to identify sick people that could be in the community. So individuals say, oh, I know of someone in that particular location who was not keeping well, and then feels like, looks like what you are explaining is what this person was sick of. And then that's how we find in these cases within the communities. And as well as also the stories on how this individual came, and then following up all of the steps, triangulating information from one phase to the other, or from one subnational to how the individual came national, or from the community onto a subnational facility, which we have our representatives there. So that is what we are doing at the moment. And then following up every single, we are not letting go any rumors at all. So we take every rumor very serious, and then it's being investigated comprehensively. And then usually it does take time, and then resources like the personnel that's needed to do that. So we have surge up, work with government, surge up a lot of the human resource to be able to cope with the alerts that are coming in, the alert volumes that are coming in over the last 3 weeks since we had the first case. Next slide, please. So basically for us as a country, that's what we have been doing, and as a program, and then as a university, we continue to work with the government, putting in place, revisiting the surveillance systems that we have that is in place, or what is needed over time, and then providing roadmap step by step what needs to be done at every point of the outbreak here in country. And we're also looking through contact tracing system, what are the feasible around the quarantining facilities, what necessary to be done over time. So that's exactly what we're doing here in response to the spillover when you're unable to really prevent it. I hand over to David, please.
Yeah, thanks, James. We're running short on time, so I know we want to get into the question session, and we'll just leave this roadmap slide there as a few points that the PREDICT team has sort of put together on what we believe to be at least a basic but an effective roadmap for pandemic prevention. But James, over to you to lead the Q&A.
Thanks everyone for those presentations. So yeah, just a reminder, you can ask your questions on the Q&A feature, and some of them are being answered on that feature as well by the panelists, so they're able, in some cases, to respond directly. Our most popular question is from Lungelo Ndlovu from Zimbabwe. How can journalists constructively communicate the urgency and severity of pandemics without contributing to panic or fear of viruses?
It's a great question. I think, James, honestly, you all are the journalism experts. I would almost defer that to your expertise. I mean, certainly we can top up on the technical side, but maybe working together with experts to identify real, true, factual-based sources of information. But I mean, presenting that information and communicating, I think falls in your wheelhouse more than ours.
That's okay. You turn it back on me. Yeah, I mean, just this is something that we try and advise journalists about at Earth Journalism Network. So please check out our website. We just posted a whole tip sheet on how to communicate, about zoonotic diseases. Certainly, you want, as journalists, you want to connect with scientists like the panelists we have here. Look for local scientists that you can talk to regularly, get their advice, and especially talk to medical experts to make sure that you're giving good tips on how to react to the pandemic without panicking. So I do urge you to check out some of the materials on our website. While we have the panelists here, let me ask some more questions for them. We have a question from Ralph Mweningwe. I'm sorry if I mispronounced your name, but David and friends, could the coronavirus that is causing COVID-19 have been detected earlier? And Mona Samari adds, are you guys surprised by its far-reaching spread? Is there anything that could have been done ahead of time to kind of maybe help prevent that from happening?
No, those are great questions. Pranav, do you want to speak to that from a data perspective?
Right. I think we, like within the epidemiology field, I think people are not really surprised with this, how far the coronavirus has reached globally. If we just look at the SARS-1 or the MERS outbreaks, which happened like in 2002 and 2013, I think they were contained really quickly just because at that time, even if we just look at the avian transportation from China globally, it has now probably increased tenfold, 15-fold within the last 10 years. So the way the virus this time spread really quickly globally was probably because of that. And of course, there are some other epidemiological reasons as well that we couldn't quickly contain the virus, but it was like within the epidemiology realm, people were scared of something like this happening, for sure.
So did we need to be checking, for instance, in wildlife markets in Asia, for instance, which you identified as a potentially at-risk interface, and is that being done either by your team or by somebody else?
Yeah, great question. There's a lot of research groups doing work in the wildlife markets in and around the world. I mean, in these wet markets, these wildlife markets are not unique to Asia.
Right.
You know, they're also pretty prevalent in West, Central, East Africa, and beyond as well. And PREDICT did do work in these markets, and we collected samples in these markets, and we worked to understand the value chain. You know, from the source animal and its natural habitat through the trade system and the network into the market itself. A lot of research groups, around the world have focused in on that, not just predicted as an obvious at-risk interface. And it's something that, requires ongoing monitoring. So, I mean, I think everyone needs to understand that viruses evolve and things change, and it's not a static landscape. It's a very dynamic process, the process of virus spillover and spread and the risks of that. And so continued and ongoing monitoring is required for something like, COVID-19. A lot of work was done in the caves in and around that area of China after SARS, to monitor, those bats, but the risks were still there, and the human animal contact with, bats in those caves, which are the suspected host reservoir species for that spillover event, are still, part of the human fabric and part of something that, we need to actively address. Basically the relationship of how humans are interacting with wildlife and the impact and the footprint of our species on the planet and ecosystems, and how that's responsible in causing some of these events. It's kind of a good message for Earth Day. We really need to work, as we presented earlier, on how to live safely with wildlife and how to live safely and together and protect habitats, and work with conservation groups and work with health experts and work with communities to really address some of the drivers of these things.
So you mentioned some of the drivers. We've talked about wildlife markets. Another questions we've had from Tracy Keeling and others is how much is climate change a factor in driving these epidemics, and as well as encroachment on natural spaces?
I think climate change is definitely playing an important role. Just if you look at the example of Flaviviruses, these are all the viruses that are transmitted by vectors, so mosquitoes and ticks, which are heavily regulated. Their populations are heavily regulated by temperature and other climatic variables. So there is a fear that these vectors will be more prevalent in temperate regions because of the global warming, and this can help this spread of viruses into regions where the populations are still naive to these infections. So it's definitely playing an important role when we are looking into the spread of viruses across globe.
Great, thank you. We've got a bunch of questions that deal with human vulnerability to specific viruses. We have questions from Parth Bandivadikar about have we identified beforehand the types of genes which are vulnerable to mutations leading to spillover infection into humans. And is it possible to determine which viruses we might be immune to, which viruses in the wild we might be immune to, and which really present more of a threat?
Right. I think that's another great question. Like, this is what, once we have discovered the new virus, this is what our lab teams are doing once we have isolated a new species. Like, for example, now we have identified the new Bumblebee virus, and the lab team is trying to understand what are the specific genes that help the virus have entry into bat cells compared to human cells, and what are the proteins due to those genes which help them infect better humans versus infect better bats. So these are all the studies that needs to be done for different viruses with different strategies to identify those key genes that are helping mutations.
We have a question from Emma Gatton. What do you think are the potentially overlooked risk areas for spillovers? Or are there any areas, we haven't talked a lot, for instance, about livestock and factory farms, so I'm wondering how much you look at that. Are there any other at-risk interfaces that we should be aware of?
Definitely livestock, industrial livestock farming is one of the important source for zoonotic diseases for humans. It's I think 8 or 10 folds higher than wildlife for sure. But and to mitigate that, like improving biosafety is essential. Improving the contact with wildlife and the contact interface between livestock and wildlife is also important. So those interfaces are very well studied, not just by our group, but by many other people across the world.
I heard you, Pranav, you mentioned that you guys had detected over 900 new viruses along with the metadata about them. I mean, that's a lot of viruses. How do you determine, several people have asked, how do you determine which are really the ones that are most threatening?
Right, that's a great question. I mean, that's why we have like a whole team of modeling and analytics. And as I was trying to show, that we try to look at viruses that we know already are zoonotic and compare them with viruses that are not zoonotic, or at least are yet to be spill over into humans. And then we try to understand what are the factors within zoonotic viruses that are making them zoonotic. They could be either ecological factors, they could be virological factors, and then try to interpret that that result on the new viruses that we have been finding. So definitely, as I was talking about the studies related to new Bumblebee virus, which is a new Ebola virus that is yet to be found in humans, the lab team is doing the whole suite of virological experiments to determine if this can infect humans. So this is how we are trying to ascertain the risk. First on the ecological footprint, whether this can be a virus that can get in contact with humans so that it can spill over. And then on the virological footprint, where we try to understand what are the specific genes that can help it mutate into humans.
Thank you, Pranav. So on my time, we're past the hour mark. I hope the panelists can stay with us for just a few more questions. And also, again, reminder, look at the Q&A feature. There are questions being asked and answered on there. So hopefully we're addressing as many of your questions as we can. Maybe this is a question for Dr. Grace, please. You have this project about living safely with bats. I wonder, if you could provide some practical advice for people who do have to live with bats. Should we just completely avoid eating bats? What do you do about the people who collect guano? Is there a safe way to do that?
So for us, I think there always been interaction. There's always been because these bats sometimes they roost in their houses. So there is a different way how we can safely live with them. You cannot get rid of them, so we have to look at the conservation partly, but again, we have to live safely. So one of the things that we tell people is that if there are bats coming into our house, there has a means and ways how to prevent, maybe to clear all the very small holes that the bats can enter in your houses. So how to renovate our houses, to build our houses that in such a way this bats cannot roost in our houses. But if there's a chance that it happens, and we have to clean up all the guano, but we have to use the protective gears in such a way. We have to put the gloves and the mask. If there is no mask, even the closed mask to prevent them. And if there's a bat died in some way, they have not to touch it directly. They shouldn't feed the animals. They have to bury it or burn it completely. But again, if it's happened in some points, they get bitten with the bats, they need to get rabid. They have to clean the wounds thoroughly with water and the soap. But again, they have to go to the hospital and get inform the healthcare workers that they've been bitten or scratched by the bats and get the vaccine. We recommend the rabies vaccine, but because some of these bats also carries the rabies vaccine. If it's not necessary really to use bat guano, especially for in our case here in Tanzania, we have so many other animals, livestock animals, where we can get the fertilized manure. It's not, I wouldn't recommend people to use the bat guano because you have an alternative. Unless for some of the countries they don't have alternative, yes, they can use. So the thing is, we have to try to protect ourselves. We have to live with them, yes, but we have to make sure that we live safely with them because they are carrying a lot of viruses and we don't know at what time the virus will spill over, because that's the thing we're still investigating. At what particular time and with it is going on with those the colonies of bats. Once we get all the answers, it's easy also to communicate more of this intervention measures.
Great, thank you. I wonder if you could talk about the COVID-19 outbreak and whether, I mean, do you think is there anything we can do to help build immunity in advance to future outbreaks, or is that building this herd immunity? We've heard a lot about building herd immunity. Is there any way we can do that short of just, you know, letting the virus run through the human population, or of course developing a vaccine?
I think people have been discussing and developing models to understand what will happen if we just allow to develop the herd immunity. But as we know that the virus is quite fatal and has significant health impacts on certain groups of people. I think people are not yet completely sure about this approach. So the best approach eventually could be to develop vaccine and develop herd immunity through vaccine. So at least to have immunity to people who are in the vulnerable group for COVID-19, and especially when it comes to emerging viruses, to avoid what's really happening with COVID-19, to avoid emergence of new viruses, we should probably think of reducing contact with wildlife and animals, and target those specific interfaces where there is quite a lot of human-animal contact that's happening, which is giving viruses a lot of opportunity to jump into humans.
Okay, and maybe just the last question, because we are running out of time. But we will try and if it's okay with you guys, we'll post your slides and presentation on our website so that people can see that. But just a last question for you. There has been a lot of talk, and you've mentioned yourself, we need to better monitor our interaction with wild animals. And there's been a lot of attention on these wildlife markets. Do you provide specific advice for how these markets should be regulated or monitored? Or do you think they should be closed down altogether, or do you provide that kind of policy advice?
I think that's a great question. I mean, I'll start and other panelists can top up. But, you know, I think the market issue and regulating the market issue it's a very complex issue. So, I mean, there's a lot of social factors involved as well and some equity considerations. You know, often, you know, livelihoods are dependent on, you know, some of these activities. So, you know, those that are engaged in, you know, in the wildlife trade, you know, that are actively part of these wildlife value chains that sort of end at, you know, at the market level. I mean, there's a lot of factors to consider there, you know, culture, income, livelihoods, and, you know, beyond just the simple blanket, you know, we can shut down a market and stop the risk. Because, you know, I think we need to be aware that, you know, regulating markets is great. Creating safer markets might be an option. We do have some partners that have spent a lot of time, especially in the Southeast Asia region, working with their host country governments on policy ideas and policy strategies around this for a long time, Wildlife Conservation Society in particular. But it's you know, not a simple context to work through. And I think, you know, when you bring in the complexity of all of the actors and all of the factors involved, it's hard to say that, you know, just regulating the market is going to stop the risk anyway. You know, there's still the risk of contact with wildlife, you know, outside of the market, you know, and you know, the risk of spillover maybe is, you know, potentially amplified by the market setting, but it's not the sole risk. So that's sort of why we've kind of taken, at least as researchers, the more value chain approach to really understand, you know, the risk, you know, along the entire, you know, stream, you know, from the wild animal in its natural habitat and into the market setting where it's finally sold or, you know, consumed or whatever. But, you know, great questions. You know, there's a lot of active work trying to really understand what the best approach should be, especially now in the COVID-19 situation. You know, I think we support regulating markets as a conservation oriented group, but getting there and getting to a specific strategy that works and that addresses all of those complexities, I think is a big challenge and something we need to keep working on.
Yeah, I guess if you just close down markets, you risk driving them underground. I know I said that was the last question. Actually, I have just one more, if you don't mind. Can you talk just a little bit about PREEMPT project? That sounds quite intriguing. Maybe just very briefly. I mean, how do we preempt, or do you have any broad comments on how we might preempt or prevent these?
Yeah. I mean, so we talked, you know, the PREDICT approach has been more kind of classic public health prevention. But the PREEMPT program is a project that's working on technologies and tools, so countermeasures, so things like, you know, vaccines and innovative ways to work around it. And, you know, the PREEMPT in Sierra Leone is, James can top up on this. He's a huge part of that team there, but they're targeting Lassa virus. And, you know, the whether or not we could potentially create a vaccine technology to address some of these deadly pathogens in their wildlife hosts in a cost-effective way. And so that team is sort of a different kind of consortium, but it's James's team in Sierra Leone, which leads the in-country activities to understand the Lassa virus ecology and doing all the sampling in the field and everything. And then a big team of laboratories in Europe and in the United States and Australia that are working to, you know, engineer some new vaccine tools that might be able to cost effectively be introduced into a rodent population and stop Lassa in the rodents before, you know, the virus spills over into people. So it's just one of the, you know, the new things on the table, that we have these, you know, excellent technologies and tons of capability, and especially when you bring all of these brilliant minds together in a collaborative way or in a One Health kind of approach type way, the way we've been doing. And, you know, I mean, the opportunities are boundless there to sort of address, and, you know, some of these challenges like virus spillover. I think we just we need to continue working on them, working together, and you know, hopefully keep advocating for the resources to do so. James, anything you want to say on Preempt in Sierra Leone?
Yes, David. In addition to what you just said, we are building One Health teams, both officials from the Ministry of Health and Agriculture, who, you know, Lassa fever in Sierra Leone is endemic, but also you have regions that doesn't have cases at all. But the reservoir and the animal reservoir is found across the country, everywhere within the country. So what we are doing is sampling, trapping rodents within the houses and out of houses in those communities, collecting samples to see whether we could find the viruses in those species of rodents that we will catch. So we've rode a year old now, multiple seasons, and then with the dry season and the wet season as well, seeing the animal abundance and then also the virus positivity rate in the animals that we will catch, and then try to see is there any different because of the environmental difference. That's why we have more spillovers in one region compared to the other region. So we're trying to answer questions, or is it that the virus strain in one region varies from the other region where we do not have frequent spillover? Or is it behavioral practices in the region where we have frequent spillover of Lassa virus from the rodents differ from the other regions where we do not have? So these are some of the questions in here the in-country team is trying to answer with the data that we're collecting in the field, and it involves engaging communities, involves administering questionnaires, and then some observational work that we do, and then alongside the rodent collection and sampling that we are doing.
Well, thank you again, all of you, for the fine work you do. It's obviously more important than ever. I can see the possibility of potentially one day providing vaccines for animals themselves, and or finding these other ways to identify risks in animal populations before they can spill over. Maybe that's the holy grail. But in the meantime, thanks again for taking the time to speak with us and to answer questions. Answers are being posted in the Q&A feature online, and once again, we will be posting a recording of this webinar on our website at earthjournalism.net, and we're having a bunch more webinars in the coming days and weeks, so keep a lookout for them. Thank you again, David, Pranav, Grace, James. We really appreciate your time, and let's stay in touch and keep up the great work. Thank you.
Thank you so much. It was a pleasure.
Thank you. Thank you for having us.
Thank you. Bye.
Thank you, everyone.